Thank you very much for that very kind introduction. The truth is, I'll always come to Birmingham if I'm asked, because you made me, so that's great. And it's a pleasure to be back here. I walked briefly by mistake, actually, on the campus, thinking I was going to Biles instead of the medical school, and so I had a quick walk round there today. So it's a pleasure to be here, a pleasure to see all of you in this lecture theatre. What I'm going to do today is talk about the work carried out in my lab, and it will have a historical perspective today, focusing on quite a few years. And I want to talk about the work from that my lab, which has contributed to the idea that cyclin-dependent kinases, these protein kinases, are the major regulators of the eukaryotic cell cycle. It's work that spans 25 years, half of which was spent actually in this institute here, which is the old Imperial Cancer Research Fund, now Cancer Research UK's London Research Institute, and quite a chunk of the work was actually done in these three bays here. I've now moved back to this institute, but they've now relegated me to the back of the institute, so I can't show you where my present lab is. It's on the other side of the building. And I'm going to talk about work, as I said, over 25 years. So some of it is going to sound pretty old-fashioned, but the reason I'm going to do that is I can, by giving you that account, give you some sense of the discovery process and the sort of trials and tribulations of going through that, even if what I'm talking about is a bit old-fashioned. And I'll try and indicate, as I go along, some bits of it which we now do really very differently, given the tools that we have now available. So this is a dividing cell. It's a dividing mammalian cell, as it happens, a fibroblast. And this is the culmination of the cell cycle. That is, the processes that lead to the proper reproduction of a cell. And this cell cycle is important for several reasons. First, it is, of course, the basis of the growth and development of all living organisms. The cell cycle leading to cell division is that basis of growth and development. Secondly, it's important for disease, particularly cancer, because cancer is a disease where cell division goes out of control and you get a deregulated proliferating mass or alteration in cell types within a tissue, and that fundamentally is a consequence of a deregulated cell cycle. And because it's a somatic genetic disease, that is, it's a consequence of DNA damage, and since the cell cycle is concerned with the orderly replication of the genome, then the cause of cancer is often related to DNA damage and not coping with that through the cell cycle. The third reason is illustrated here. This is the cell cycle. It's what I call the stripped-down version of the cell cycle. Let me say something about that. If you read a review of the cell cycle these days, you will get about 2/3 of the way through the review, and they will produce a summary model, and it'll have about 100 genes on it, and all it does is depress you, you know. So I always start with a simplified problem, because you have a statement of the problem, because it encourages for you to feel you might solve it, actually, whereas the other way you don't. But the reason I put this up here is that it emphasizes that in each cell cycle there are two major processes: DNA replication, when the genome replicates, followed by mitosis, or M phase, that you see up there, when the replicated genomes are segregated into the two newly dividing cells. And the reason I wanted to put this up was not only to remind you of the S and M and the G1 phase and G2 phase, as I should be talking about these things, but also to point out that what the cell cycle does is takes the double helix and the manner in which it's replicated and turns it onto the stage of the cell. Because we of course know about the importance of the double helix and replication of the genetic material, but that will only matter if it's actually in the context of the cell and in the context of a cell dividing that ensures the proper segregations of those genomes. And it's the cell cycle that turns the molecular behavior of the replication of DNA into something that means something cellular, and that is biological. And I think that's the third reason, one that isn't perhaps mentioned so much, which is important. Now, this process has been my obsession for now nearly half a century, which of course means I'm rather a sad, emotionally underdeveloped person. That's certainly what my family think. But this is what I have been obsessed with trying to understand how this process works. And I'm going to take you through some of the key experiments, some of them going back to the 70s, which led eventually to our contributions on the models of cyclin-dependent kinase. Now, I've taken a genetic approach to this problem, and I used a model system, the fission yeast Schizosaccharomyces pombe. This is it. It looks like an overgrown Escherichia coli. It's a rod-shaped organism. It's a pretty useless yeast. It's not useful like budding yeast. It doesn't make any decent wine or bread or beer. It makes disgusting beer. I have drunk it actually, and it is truly disgusting. But it turned out to be quite a good model for the cell cycle. It's a simple eukaryote. It has the basic properties of the eukaryotic cell cycle. It has around 5,000 genes. My lab led the sequencing of it. It was the fourth eukaryote sequence, actually. And so it's a simplified system for looking at this problem. Now, the story of my sort of working on this began when I was a graduate student. I left Birmingham in 1970, and I went to the University of East Anglia. Your present Vice-Chancellor, was Vice-Chancellor of UEA before he came here. And I did my PhD in Norwich between 1970 and 1973. And I was working on amino acid pools in plants and in fungi. And like all graduate students, I thought I had the worst project in the world. The only difference is that, in my case, it was true. It was fairly tedious. I had actually a very good supervisor, but the work I was doing it turned out not to be terribly interesting. And central to it was a machine that had just been invented. This is 1967. It was an amino acid analyzer. Some of you in the audience might remember these things. And what we had in the lab was that dreadful thing which is a machine prototype. Machine prototypes have one common characteristic, which is that they don't work properly. And this was the case for the amino acid analyzer. And I had to analyze amino acids out of various pools. And it had a very interesting quirk. It took to run a profile 2 hours and 42 minutes. I still remember it all these years later. And it had a very nice design feature, which was this, that it collected all the data, and if it went wrong, switching itself off before the end of 2 hours and 42 minutes, it dumped all the data that it had collected up until that time. And given that the machine worked on plumbing, because it did chromatography, and it had too many pressure safety devices, it switched itself off about 2 times every 3 runs. And so you threw away all your data most of the time. Okay? Now, Beckman said they were going to iron it all out, but I had a PhD to sort out, and it was just too slow. So I took what all graduate students faced in this awful situation, as we often were, the usual approach, which was to go through the machine and inactivate all the safety devices that I could find. This was not done by any clever sort of electronics or anything. It all involved sort of rubber bungs, sellotape, and sort of blue tack, until I had a machine that would often get to the end of 2 hours and 42 minutes, but was actually rather dangerous, because it was now operating most of the time about twice the safety pressure that it should be operating at. So given that, and this is all relevant, I know it sounds utterly irrelevant, but it is vaguely relevant to what I'm going to say. So this meant to actually run it, I had to sit with it, okay? And I had to monitor the pressure gauge, so if it really got dangerous, I switched it off, okay? And this is, of course, extremely boring. And so what I did is something else that graduate students often don't do, which is read papers. What graduate students do on the whole is print off papers or photocopy papers and put them in heaps on their desk, but they don't actually read them. And this, giving me 2 hours 42 minutes in slots, meant that what I used to do is hold the paper like this, and I had my finger over the stop button, and the pressure gauge was in my peripheral vision, so that if the pressure went up too high, I could stamp on the stop gauge. And I read paper after paper after paper. And I read some papers then by a character called Lee Hartwell. And Lee Hartwell was a budding yeast geneticist who was just starting, this is like 1972, 73, on a genetic analysis of the cell cycle, isolating mutants that were unable to complete the cell cycle. This was abstract genetics, there was no molecules in sight, it was just identifying genes. I read it and I thought, this is quite cool. Cell cycle was an important problem. I didn't say cool then, by the way. I mean, you only say cool now. But it looks a very interesting piece of work, and I was quite attracted to it, and I read very carefully to see if there was any mention of a Beckman amino acid analyzer, and I saw no mention of that, so I thought this could be something that I would be interested in. The problem was, although I had learned very well genetics in Birmingham, and I have even my old lecturers here today, and even though I had learnt genetics, I had not done it as a PhD student, so I had to go and learn genetics to apply a genetic technique, and then use it in a lab working on the cell cycle. So I looked for somebody working with fission yeast, which I thought would be a better model than budding yeast, and I found the man on the left there, called Murdoch Mitchison in Edinburgh, who had started working in the 1950s on this problem. Here he is on a Burns night doing the washing up. You can tell it's in Edinburgh by the kilt. He worked in a kilt. That's my wife behind there. Can you see? Whenever she sees this photograph, she always says, we're still married, by the way, which is something else I suppose fairly extraordinary. But she always said, look, we're doing the washing up and Paul is nowhere to be seen. But this is Murdoch, and I went to Urs Leopold, who, as you can see, lives in Switzerland, or used to live. They both died, these two gentlemen, with an alpine horn. This was his 60th birthday. And I went to Leopold. Leopold taught me genetics, and I spent 6 months with him. And then I went to Murdoch, and I worked with him for 7 years. And what I wanted to point out is that I published 15 papers or thereabouts during this time, and they didn't put their name on any paper, not because I think they thought it was all rubbish, at least I hope not, but simply because they didn't feel they'd contributed sufficiently. In the case of Murdoch, he hadn't contributed with his own hands. We don't do this really very much anymore, and I just want to acknowledge they both, as I said, have died. These were true gentlemen, as we would say, of science, and extremely generous, and I'm very grateful for the help they gave me at this time. So, the first thing I did was isolate a load of temperature-sensitive mutants that couldn't grow at a high temperature, screen them for mutants that were unable to complete the cell cycle. I did this work with two other individuals. Kim Naismith was a graduate student who worked with me, now Professor of Biochemistry in Oxford, and this is him then. And what we looked for, A is a fission yeast dividing cell with that white line in the middle showing dividing cells. B, C, and D are all CDC mutants, cell division cycle mutants, unable to divide, but still able to grow. Because obviously if you stop a cell making protein or RNA or energy, they will stop dividing, but that's a trivial reason. What these mutants do is stop the cell reproducing itself, even though they can grow. So that's the way into the problem. It had identified genes necessary for cell cycle progression. At this stage, in this paper here, in the 1970s, I identified 30 genes with my two colleagues here. We've since deleted every gene in fission yeast, actually, and screened for all the mutants that are involved in the cell cycle. We now know there's about 400, between 300 and 400. So at this stage, we had 10% of the total number. But we had a lot of luck to get some interesting genes in that particular collection. Now, the logic behind this is shown here. What we see here is, and I'm using the illustrations I drew at the time, just to sort of, so this is all pre-PowerPoint and all that stuff. This is my handwritten hand-drawn stuff. And what you see here is a normal cell. Can you see the normal cell cycle? It's a bit difficult for me. Oh yeah, I can do it here. And it grows, its mass increases, it divides, and it goes through this cycle. And if we're blocking an event necessary for proper reproduction, you get this CDC phenotype that I've just showed you. Now, the reason I put this up there is for a different reason, however, because I told you at the beginning I was actually interested in the control of the cell cycle. I was less interested in genes needed for, genes necessary for, and more interested in genes that control. Now, control is one of these words that means different things to different people, and the definition I was thinking of here is a particular one which I'll describe, which is I was interested in genes that would identify functions that acted as major rate-limiting steps for cell cycle progression. In other words, weren't functions that were needed for cell cycle progression, but were ones which, if you did a thought experiment and lifted up the activity a little bit or turned it down a little bit, it would influence the final timing of cell division. I was very much influenced by analyses of metabolic flux through metabolic pathways that was going on at the time, both in Edinburgh and Oxford. Henry Catcher, for those that might remember, was one of the people who was doing that, and who identified rate-limiting steps in metabolic pathways. And it's in exactly the same logic that we're talking about here. Now, my thinking was that it is relatively easy, if you knock out a function, to make it rate-limiting. Because if you take a function that's 100%, you knock it to 0%, it becomes rate limiting. More interesting is if you can change it a little bit. In theory, if you change it from 100 to 70 or 100 to 50, if you have a significant effect, then it's going to be much more interesting. But it is easy to block cell cycle progression, much more difficult to speed up cell cycle progression. And if you can speed up cell cycle progression, then it's much more likely that you've identified a rate-limiting step, because you can't speed up a step which is not rate-limiting. Whereas if you speed up a step that is, then it will be. And so now we go to the other part we have here, which is we have a normal cell cycle proceeding here. And if we now speed up a function that's rate-limiting, and so you undergo the cell cycle more rapidly, instead of waiting to here, you will actually divide, let's say, here, and the cells will divide at a small size rather than a large size. Dead simple. A small size if you're advanced through the cycle. Now, this then potentially identifies rate-limiting steps. Now, I'd like to explain to you that I sat down one day and had this thought, and then went and looked for these mutants, which incidentally I called wee mutants because I was working in Scotland at the time, and it seemed quite funny. I have to say one thing, is if you are still naming genes in the audience, there's a great temptation to think of funny or cute names. Before you do it, just remember you may be talking about it 1 year later, 10 years later, or in this case nearly half a century later, and seldom does it remain funny for that length of time. Now, I'd like to tell you that I thought this and then looked for them, but of course I didn't. I wasn't thinking this at all. I was actually screening visually under the microscope for long cells. And I'd centrifuged them through a gradient to get long cells, okay, big cells, through a sedimentation velocity centrifugation. And whilst I was visually screening, I saw a patch of small cells, just as one would, you know, at the bottom of a gradient, where, of course, they should be at the top of a gradient. And I looked under the microscope, I saw these small cells, and then I constructed the logic here after the event, okay? So the point I'm emphasizing there is serendipity, following, as I said to some of the students and postdocs a bit earlier, where nature leads you can be extremely powerful. I hadn't thought of this, and it basically opened up this pathway, and here is the first mutant. I called it WE1. It was published in 1975. Notice sole author, because Murdoch and Ers said, just go for it yourself, okay? I was helped a lot in discussions by my colleague Peter Fantes at the time, and you can see this is wild-type cells, this is the wee mutant. The first mutant I got serendipitously was temperature sensitive. At low temperature it looked like this, at high temperature like this. This allowed me to shift from low to high temperature, and then ask the question, where in the cell cycle does it act? I don't want to go through the details of that, but the reason you can do that is you shift at time 0, you can ask how long did it take before cells start dividing at a smaller size, and that will tell you where in the cell cycle it acts. You can see intuitively, I'm sure, how it works, and you can see here the size at division dropping and a cell division being accelerated, and that allowed me to conclude that there was a rate-limiting step in the cell cycle, because there didn't have to be. Secondly, that We1, which is what I called this gene, was important for it. And thirdly, the cell cycle timing suggested that it acted at the G2 to mitosis transition. So the major rate-limiting step in this cell cycle was G2 to M, out of this simple experiment. No cloning, no nothing, okay? Just getting a mutant and doing simple experiments. So what I then did, having sort of been given this on a plate, was thought, right, I can genetically analyse rate-limiting steps by looking for Wee mutants now rather than CDC mutants, okay? Now these are tedious to get because they grow, so all you can do is mutagenise and visually screen, okay? And I visually screened, and it took me about 18 months to gather 50 mutants, okay? And the first thing I wanted to know was whether any of these mutants, and most of them mapped to a single gene, to We1. I'll tell you about one that didn't in a moment. 50, but most of them mapped to a gene called We1. And because it was so frequent, that made me think that it was a loss of function that generated the advancement, simply because it was easy to get. But that wasn't very good proof. So I undertook an experiment which now seems terribly, terribly weak to try and improve that. But I'll tell you what it was and to show you what pathetic tools we had at the time. Because now all we do is clone and sequence it, and that'd be the end of it, okay? And you do it in about 2.5 minutes. But then it wasn't like that. So what I reasoned was this, that if I could discover a mutant that was a nonsense mutant, therefore prematurely terminated, right, that is most likely to lead to a loss of function, is what we reasoned, because you only had part of the polypeptide. Now I actually know that it's not true anymore, but at the time it was thought to be the case. So what I thought we would do is take all these mutants and cross them to a panel of nonsense suppressors, that is tRNAs with a mutation in the anticodon site that would read through a nonsense mutant. This is real old-fashioned tRNA nonsense genetics, and Urs Leopold was an expert at it, so we had all those mutants available. So I took those 50 mutants, I crossed them to 9 or 10 suppressors, and went through the whole lot to see if any of them was a nonsense suppressor, and one was, and that was We1 112, if I remember rightly. As you can see, this was published 33 years ago, and you can see there that the cell length at division, SUP35 was a suppressor, in a wild type is 14.7, in the 112 mutant it was 7.5, and it was nearly fully corrected by SUP35 to 13.1, okay? And so I published this as showing that We1, because of the frequency of mutation, and it was a nonsense mutant, was an inhibitor of the G2 to M transition. Terribly contrived argument, and you wouldn't even get a look at it if you'd published such a thing today, but it worked, at least in the journal Genetics. Now, I told you that I had 50 mutants, which I got through. 49 of them mapped to We1. One did not. This one was actually the 47th or 48th mutant. I gave myself 50, I would isolate. And I remember it very, very well. I was in Edinburgh, and I found it about 5 o'clock on a Friday evening, and it was raining, and it had an aspergillus contaminant on it. And I don't know if any of you who know that fungal contaminants are the kiss of death to a microbial geneticist. And I was fed up, I'd been working all week, and I threw it in the rubbish bin, okay? And I went home, and I had my tea, and then I felt guilty about it. And I remember cycling back in the rain and retrieving it from the rubbish, okay? I got it out of the rubbish, and you know what you do? You cut all the fungus out, you pick up a little bit, you put it into the plate. I did about, over the next week, about 3 subcultures. In the end, I cleaned it up, okay? I just thought it wasn't worth the effort. It turns out that that mutant did not map to WEE1. Okay, the only one of the 50 that did. It mapped to a second gene that I called WEE2. At this point, I realized my name was getting in the way of the investigations, okay? So I now had WEE1 and WEE2. Now, what I reasoned was that if I was really lucky, since WEE2, I should say, was rare, and when I made a heterozygous diploid, it was semi-dominant, okay, to dominant, it was possibly a gain of function activity rather than a loss of function, right? And if it was a gain of function, that is to say accelerated activity was leading to acceleration through G2 into mitosis, then if I knocked it out, it would block division and give a CDC phenotype. And therefore it was worthwhile asking whether WEE2 was allelic to any of the 30 CDC genes that I'd already identified. Remember, that's less than 10% of the total genome. So my second piece of luck. WEE2 turned out to be closely linked to a gene I called CDC2. And so I couldn't get, I had less than a centimorgan recombination between them. So it was potentially an allele of CDC2. But I was worried, and I'm quite anal, that actually it wasn't CDC2, but just a gene adjacent to CDC2, or close by. So then I thought what I have to do to prove this is to isolate more CDC2 mutants and do a fine structure map by classical genetic mapping. Again, rather like Benza did for phage. So I went back to the screening again, spent another year isolating many, many CDC mutants, and identified about 10 to a dozen, I think about 10 new CDC2 TS mutants, made a fine structure map. I don't know, you don't want to know the details of this, but I can tell you this is recombination levels of between 10 and 100 in a million, so this was a lot of work, and I constructed a fine structure map, which is seen here on the left, and I then mapped it with respect to the WEE2 allele, and the WEE2 allele was within the CDC2 gene. All I can say, by the way, is thank God for this allele out here, otherwise I might still have been worried about it. Very good was one TS allele, CDC256, I can't see where it is anymore, but it's somewhere up there, was temperature sensitive and WEE at low temperature and CDC minus at high temperature. So that confirmed it. That I now had 2 genes, WEE1 and CDC2, and WEE1 was a negative inhibitor of G2 to M, CDC2 was a positive activator of G2 to mitosis. So at this point, I felt we'd identified a major rate-limiting step in the cell cycle, fission yeast. It acted at G2 to mitosis, regulating the onset of mitosis. We had 2 components, an inhibitor and an activator, right? I was excited by this, okay? The problem was, nobody who might be offering me a job was excited by it. And this is something that's familiar, I'm sure, to many in the audience. I would come and talk about this. It wasn't trendy or sexy at the time. It was a lot of classical genetics and physiology, and people just weren't interested, okay? Those that were interested in the cell cycle said all the regulation is anyway in the G1 at the beginning of the cell cycle, and so this is just something very peculiar. So I couldn't get a job, okay? So this was troubling, because I was looking for a job. And I thought, what I should do is see whether there is any, what sort of controls acting at the G1 to S transition, because that might help me get a job. This is serious. I thought I would do that, okay? So I went back and talked to who was now my friend Lee Hartwell, who was working in Seattle. And what he had done was a very clever experiment. Oh, by the way, this is Pierre Thurio, my friend here. This is me, incidentally. If you hadn't recognised me, I should have pointed that out. I had hair and colour then, and looked a bit like Frank Zappa. And, you know, you can just see what looks like the drug fumes coming off the top of his head. And he helped me with this study here. Now, this study, though, was done with my technician at the time in Edinburgh, Yvonne Bissett. And it went a bit like this. It's a complete copy of what Lee Hartwell did. What Lee Hartwell did was to use his CDC mutant collection, I'll remind you, in budding yeast, to determine the point in the cell cycle when a cell becomes committed to the mitotic cycle, right? And he used a sort of developmental genetics logic to it. What he sort of said is, I'm going to block cells at different stages in the cycle with the CDC mutants, and then I'm going to challenge them to do something else. If they can't do something else, then I'm going to argue they are committed to the mitotic cycle. If they can undergo an alternative developmental pathway, they're still flexible enough to be diverted from the cell cycle, and they're not committed. Now, the only thing, yeast being rather dull organisms, the only thing they do that's different is, in fact, to conjugate and undergo meiosis. They have a sexual cycle. So what he did was to challenge cells blocked at these different stages to undergo mating and conjugation and by a mating assay. And he had identified a point in G1 he called start, where cells became committed to the mitotic cycle. So I thought, I've got the CDC mutant collection, I can do the same assay, I can see it as a start control. If there is, maybe I'll get a job. Okay? That was the sort of logic behind it. And I did the experiment, and it worked pretty well initially. Okay? So I blocked cells at different stages. There's a collection of CDC mutants that I did. I used a collection of mutants. I looked at ones in G1, and I found a gene called CDC10 blocked where there was very high levels of conjugation. I haven't looked at this figure for a while, but you'll see figures like 100% up here. And so that was good. I then looked at mutants blocking later in S phase, G2 and M, and they all had a much lower conjugation. So I had in my head, not being a sort of a numerate biologist, that sort of anything over 80% was 100%, and anything below 5% was 0%. So I could turn it into either you conjugate or you didn't. I had the whole thing ready to submit. But I thought I'll do a control. Big mistake to do control. Okay? I'll do a control, and the control I will use is CDC2, because that's the one that I've characterized most as blocking at the G2M boundary. And I did it last. And I did the experiment, and I got 25%. And even I had trouble persuading myself that was less than 5%, or which is what it should have been, but it obviously wasn't 100% either. So I assumed I'd made a mistake. I did it again, and I got 22%, or something close to 25%. I then did what all biologists do when they have this sort of problem working with temperature-sensitive mutants and so on. I bought a more accurate thermometer and set the water bath again and did the experiment again, and 25%. Okay? I got a bit depressed at this point and put it all away and fiddled around in the lab as one does in something else, and then opened it up again 4 or 5 weeks later, did the experiment again, 25%. Okay? So this was a bit of a problem for me because my control wasn't working. Okay? And everything else had worked, and I was ready to submit. And then I had a dream. And in my dream, I woke up and I had an angel on one shoulder and the devil on the other shoulder. And the devil was talking to me in my dream. And the devil said, you've got all this data together and it all works except for the data with CDC2. And you know something? The only person in the world who knows about that data is you. And you could publish the rest of it and forget about CDC2, and you'd have a perfectly good paper. It would show there was a G1 control. The world would be interested in it. You would get a job. Your 2 children wouldn't starve on the streets. You'd be able to pay the mortgage, and all you have to do is to forget about that little bit of data. Then I had the angel on the other side. He said, I'm sorry, Paul, you can't do that. Science is the pursuit of truth. You cannot hide the data. I know that you won't be able to publish this paper. I know that means you won't get a job. I know that means your children will starve and you will be living on the street. But that is just it. You have to pursue truth. And that's all you can do. I woke up in a cold sweat. And anticipating Tony Blair, I thought there has to be a third way. And the third way, and this is now moving closer to reality, the third way is something you won't believe that I hadn't thought of before. What if the data was correct? I had for the previous several months assumed the data was simply wrong because it had not turned out how I thought it should be. But what if the data was correct? And I hadn't given that a second thought. How could I get hold of an answer like 20 to 25%? So I thought about it for a day or two, and I realised there was one very unlikely explanation, and that's shown up here. I'd shown CDC2 operated at the G2M boundary. It turns out in the fission yeast cell cycle, G1 is extremely short. So if there was a second role for CDC2 here at this start role, if I shifted it to the high temperature, some of the cells would be blocked before start. The majority would be blocked before mitosis. And maybe I was sampling simply the cells blocked before start, and actually CDC2 was acting at the two major controlling steps in the cell cycle, one in G1 and the second in G2. I thought this was very unlikely, but I could test it by synchronizing all the cells in G1 and then shifting to the high temperature and asking if the cells were blocked in entry into S phase, which would show there was a second block point. Okay? And I did that experiment. It's a crude one here. It was before the fax, if you can imagine worlds before the fax. And I had to measure, and there's some old biochemists in the audience, by diphenylamine, that some of you might remember, that sort of thing. And this is diphenylamine estimates of DNA. This is the wild type control, it undergoes DNA replication. This is the CDC2 mutant, shifted up from G1, it did not enter into S phase. So this experiment, taking the data seriously and then testing the hypothesis, established that CDC2 was required at the two major controlling points in the cell cycle, at G1 to S, where it was the point of commitment to the mitotic cycle, and G2 to M, when it was the major rate-limiting step through the cycle. So with that paper, I then thought that this is an interesting gene, and this would be the key to understanding cell cycle control. Now, I've spent most of my talk talking up to here because I thought this would be interesting and possibly entertaining. I'm now going to compress the next bits, which are more conventional, you'll be pleased to hear. So it doesn't go on like this for hours and hours, is what I'm trying to say. Because the next stages was really, what does this all do? I've got W1, I've got the rate-limiting steps, controlling step, W1 and CDC2. What is it that they actually do? And this work, I've been working in Edinburgh, and I then moved to Sussex, I'll tell you why in a moment, then to London, and then to Oxford, and there's various collaborators that I've listed up here. And the first thing was required was this. Everything I've described to you has been an abstract analysis. I haven't mentioned a molecule, right? That's because we had no way of finding molecules. And the way you can find molecules in simple systems like this is today, but wasn't then, is to be able to transform the yeast with a library and clone the gene that encodes the defect, sequence it and analyze it. But we couldn't, this was 1979, 1980, cloning had only just been invented and got out there, and we couldn't transform fission yeast. So the first thing I had to do, and I stopped working on the cell cycle for a couple of years, and thought I have to develop molecular genetic techniques for fission yeast, and developed high-frequency transformation. I did that with a postdoc in my lab, David Beach, and that allowed me to clone genes by complementation. If you don't know how to do that, I have a slide in 5 minutes that will explain that. Basically, you clone it by rescuing the mutant phenotype. Now, I told you I work with fission yeast. There's very few people who work with fission yeast, and I wanted to convince the budding yeast community that I was doing something interesting. So what I did was, I cloned the CDC2 gene by complementation from fission yeast, and at this time, I had it on a 2 kb fragment, okay, kilobase fragment, and it took about a year to sequence it. Furthermore, it was a specialist activity. I contacted Fred Sanger. Fred Sanger put me in contact with one of his postdocs, and he said he would sequence this gene, but it would take him 12 to 18 months. Okay? 2 kilobases. True. Just did it. Okay? Do it in about half a second now. Okay? And whilst that was going on, I thought, can I make a link to budding yeast? And this is where serendipity helped again. Because I realized that if I could find an equivalent gene from budding yeast, then that would suggest that the control was conserved. So how can I get an equivalent gene from budding yeast? So what I did is I took a budding yeast library and transformed it into fission yeast to see whether there was a gene from budding yeast that would rescue the CDC2 mutant. I got a clone out that rescued it, so I had a DNA fragment which would rescue. But the question is what gene it was. Well, at this stage, there had been 4 CDC genes cloned in budding yeast. That's just 1% of the total. They've been done by somebody, Lee Hartwood's postdoc, called Steve Reed. So I thought it's worth a punt. Is my gene that I cloned the same as any of those 4? I mean, what, talk about a chance. Did sudden hybridization, and it hybridized with CDC28. In other words, CDC28 in budding yeast was the same gene as CDC2 in fission yeast. These are highly diverged organisms. And furthermore, CDC28 was the start gene in budding yeast. Okay? So at this point, early 1980s, we'd now established that the same control seemed to be operating in budding yeast. After that, we had the sequence. So I put the sequence into the database, right? There was about 150 genes have been sequenced at that time, and the majority of them was cytochrome C, okay? So there was very, very little to compare it with, and out popped a sequence similarity with SARC-P60. SARC-P60 is an oncogene, and it's a viral oncogene, and it was thought to be, at 90% but not 100% at the time, a protein kinase. A protein kinase is an enzyme which phosphorylates other proteins, putting a phosphate on, changing their function. So I guessed that CDC2 might be a protein kinase. The sequence similarity was small, but it was the only one that picked it up. So that was proved to be the case. I did that, you see there on the paper on the left. Viesta Simonis, postdoc in my lab. What we did is we took the CDC2 gene, we expressed it in bacteria, we made protein, this was quite early on for these sorts of things, injected it into a rabbit, got an antibody, and then started doing immune precipitation assays from fission yeast cells to assay for protein kinase activity. I have to say, I proposed this to the MRC to get a senior fellowship, and it was a very entertaining interview, yet another time when I was desperate for a job. And because what happened was there were 2 people who, both of whom I respect enormously, on the interviewing panel. Sidney Brenner, who will be known to many of you, who was rather attracted to this proposal, and Marty Raff, who is the immunologist who knew about this, who thought this was bunkum and would never work. And so Marty started to attack me over it. And then Sidney started to attack Marty. And then I was suddenly irrelevant for this. And they just argued for about the next 10 minutes about this. And I just sat there in complete silence. And the end of all of this is that they funded me. So it worked, and I wanted to show you this here. We got a substrate against casein. It's just, this is a milk protein, which I mean is irrelevant to this thing, but it just happened to work. And what I wanted to show you is to show, I'm a geneticist, and the idea that you immune precipitate this gunk and then you get P32 incorporated just terrified me as if it meant anything, And so the one thing we added to it, which was borrowed from the phage people, was to make extracts from TS mutants in CDC2 and then assay them in vitro at the low temperature and the high temperature, and show from in wild type it wasn't temperature sensitive, and in the TS extracts of a TS mutant it was. And since the mutant was TS for CDC2, the only thing that should be TS in there was CDC2, and the protein kinase activity was temperature sensitive, and so that showed whatever stuff we were assaying was due to CDC2. So that allowed us to assay it, and then we could get better substrates. We've got H1 histone as a substrate. This is me again, by the way, now looking like Robin Williams. And this is Sergio Moreno, my postdoc, who showed by synchronous cultures that the CDC2 protein kinase, shown here, peaked in late G2 just before the onset of mitosis. So at this point, we could conclude the major rate-limiting step in the cell cycle was in G2M involved a protein kinase, CDC2, and it was activated late in G2 to bring about mitosis. So we had a sort of molecular explanation. We could also put together some regulators. I'll go through all of this quickly. We1, I told you about, we showed by genetic analysis and molecular genetic analysis, seemed to work upstream of CDC2. We cloned it and showed it was a We1, was another protein kinase. CDC25 we showed was acting in a counter way, and we postulated it was a phosphatase, CDC25, which it turned out to be, though we didn't prove it. I have to say that I speculated on it because I got stuck on a Boeing 747 without any reading material before you had films, you know, when you just sat there. And the only thing I had in my bag was CDC, the sequence of CDC25, and a couple of phosphatase sequences. And I stared at these for about 7 hours, and I discovered 3 amino acids which were in common, quite rare amino acids that were in common. And I really thought about it, and I thought, I can't take this seriously. And they turned out to be the residues in the active site of the phosphatase. You know? But we couldn't take it seriously. So we postulated this little control network up there, and Kathy Gould came to my lab from Tony Hunter, and did some proper biochemistry, and took the CDC2 protein, after labeling it with buckets of P32, 25 millicuries, ghastly amounts of P32, sequenced the protein, identified the amino acid residues. It was relatively simple, thank goodness. A threonine 14, a threonine 167, a tyrosine 15, and the main thing I want to show you, these were very, very difficult experiments, was that in a CDC25 mutant, which is defective in the phosphatase, as you can see here, CDC2 became dephosphorylated on the tyrosine here. Oh yeah, no, sorry. CDC25, tyrosine was phosphorylated, but when we released it, then it became dephosphorylated. So that suggested that this model here was biochemically probably correct. Now the final stage. I'll skip that. So at this, well, we'd also cloned another gene by Ian Hagen, graduate student in my lab, and this was a gene called CDC13, and this was similar to a gene that had also been cloned by my colleague Tim Hunt, who had identified a protein called cyclin in marine invertebrate, I think a sea urchin at the time. Now the question for me, so this was where we were then, that the major rate-limiting step G2M, that CDC2 interacted with a protein CDC13 that we thought was a cyclin, and it was regulated negatively by the We1 protein kinase, and countering that was a CDC25 phosphatase. Now the question was all of this or any of this relevant to humans? Okay? And I realised this was an important question because I just moved to ICRF, not only because it's an important question, but for the following reason. ICRF at the time, this is the 1980s, worked entirely on tissue culture. And when I turned up working in yeast, I suddenly found everybody in the coffee room sitting at the other end of the coffee room. And they, as far as they were concerned, all I was a source of contamination. And so I thought I ought to really see whether any of the genes we've identified, particularly CDC2, is found in humans, okay? And which would validate this as a universal model. And the person who did this was a postdoc in my lab called Melanie Lee. And she came to the lab and said she wanted a difficult project. So I said, well, fine, if humans have CDC2. Now, this was quite difficult because you'll appreciate that humans and yeast probably diverged 1.5 billion years ago, something like that. Just in case some of you are not so familiar with evolutionary timescales, dinosaurs went extinct about 65 million years ago, so it's just a pick of time in comparison. So it's very, very ancient at the origins of eukaryotic life. So Melanie came into the lab and she did conventional ways of trying to clone a CDC2 gene, which was based on structural similarity. That is, we looked by low stringency southern blotting to look for hybridization between genes, and we used something, again, some of you will be familiar with lambda GT12, I think it was called, expression libraries. We expressed in bacteria and used antibodies against CDC2 to see if we could find the gene. Now, we did that for about 18 months, and we found protein kinases. But the problem I had is, with 1.5 billion years, by the way, no organism has been sequenced at this time. So don't just say, why didn't you go to a database? Nothing had been sequenced. 1.5 billion years divergence, how would you know whether a gene was similar or not? You know, because there was sequence similarity, but what would you need to find it? And so I got frustrated thinking, well, we're getting protein kinases. I've no idea if they're the right protein kinases. So we shifted to the same approach I showed you with CC28. We took a human cDNA library, which I got from Okiyama and Berg. It was the only cDNA library that was any good. It was expressed behind an SV40 promoter, and I knew that SC40 worked in fission yeast because I'd done some work on splicing in the meantime. So I knew if we took this library, it would work in fission yeast. I mean, I didn't have to do anything with it. So we took the human library, which is a very good one, we transformed it into a fission yeast cell. This is cloning by complementation, where CDC2 was TS, so the cells all died at the high temperature, right? And the notion was, if there was a gene in the library which not only was structurally similar, but functionally equivalent, that's the key word, then that could rescue, if that rescued this function, so that we got a CDC2 substitute, then this would now rescue the mutation and you'd get colonies on a plate. Then you could go back and isolate the cDNA out of it and sequence it and see if it's the right thing. Now this again was a bit of a stretch, you know. First of all, that the gene existed. Secondly, that it was in the library. Thirdly, that it would work. Okay? Really had no reasonable thing for working. Well, we got transformation. We got clones that grew. This is actually, I took a photograph of the very, very first cells that we saw growing. You see the guys with the long cells there, the long ones? They were because the plasmid was being lost because it was on unstable plasmid, and the others were growing. Now, this was now in the mid-80s, by which time you could sequence a bit faster, and we could sequence in the lab. So the next thing we had to do was a whole series of experiments, including sequencing, checking it wasn't a suppressor, checking it wasn't a contaminant, all that sort of stuff. And this took about 2 or 3 months. And I've got to tell you, it was quite a peculiar time for me. Because I knew that if this experiment had worked and we got human CDC2, this meant that the cell cycle control was the same in all eukaryotes. And it would also mean that, that you could use organisms like yeast to study basic problems in organisms such as humans. So I knew it was quite a big deal. But I was convinced that we were being misled. You know, we had a contaminant, we had a suppressor, we had all of this. And every time we went in, you know, a few days, Melanie and me, we did it together, it passed the next test. And I was sure I'd go in the next day and it would fail. You know, we'd do a test and it would fail. And I started going home in the evening thinking, I'm going to imagine that it's worked. Just so I can enjoy it, because I'm sure tomorrow morning when I go in, it will have failed. You know, science does funny things to people. It really does funny. I'm going to imagine it's worked so I can enjoy it, okay? Because tomorrow morning I will go in and the colonies won't be there, or the sequence will be wrong, or something will go wrong. Then eventually we got to the point where the sequence came out of on the computer screen, you know, the matching, and it was 61% identical at the amino acid level. I can tell you I went completely ballistic. Melanie, who's altogether, as you can see, a more controlled-looking person, was much calmer, but I was utterly out of control. This is it. This is the sequence identity, highly conserved. On there, you have to read the sequences. It's budding yeast, human, mouse, chicken, fly. We added plants a bit later. So it's found reasonable identity across the board, and they are functionally equivalent where we've actually tested them. Second experiment, just to confirm it, was to show that actually CDC2 was a component of an activity called MPF, or maturation promoting factor, that had been discovered in Xenopus in the 60s, actually, and it had been purified by Jim Maller, who's up here on this joint paper, and my postdoc Chris Norbury used our antibodies from fission yeast and human to show this is MPF, which was discovered by Yoshio Masui, who got the Lasker Prize for this. And then we showed that the purified MPF done by Jim Maller, shown here, cross-hybridized to the antibodies we'd prepared against the fission yeast and human CDC2, showing maturation promoting factor. What it did was to, if you injected it into oocytes, it would advance them into M phase. So it was a sort of biochemical equivalent of a rate-limiting step for M phase. It was meiotic M phase, but it was an M phase. So that showed that MPF isolated biochemically was the same, contained the same components as we had identified genetically through yeast. So that finally confirmed it. I got excited, as you can see, dancing with Marcel Doré, where we showed it in starfish and stuff like that. And so we come to the final conclusion of this, which was that there is indeed a universal control, which is rate limiting for, in this case, the onset of mitosis, and that involves CDC2 complex with cyclin. It's regulated by WEE1 and CDC25. Cyclin is degraded at the end of mitosis, as Tim Hunt showed, and the activity of CDC2 is activated during M phase and brings about the processes of mitosis, either indirectly or directly. In eukaryotes, there is a more complex set of cyclin-dependent kinases. I wasn't involved so much in this. There are indeed cyclin-dependent kinases working at G1S. In fission yeast, it's the same one. We can simplify it so it's the same one. In mammalian cells, there is a range of CDKs operating at different stages of the cycle. Now, I think it was a bit unexpected that such a complicated process as the cell cycle would end up being regulated by A, such a simple system, and B, would be common from yeast through to human beings. But I'm going to end my talk on a quote from Theodor Schwann in 1839, which, who knew better. This is what he said. We have seen that all organisms are composed of essentially like parts, namely of cells. This is a statement of the cell theory. Then look at these next sentences. That these cells are formed and grow in accordance with essentially the same laws. Hence, that these processes must everywhere result from the operation of the same forces. Exactly anticipating what we took 150 years to show. Thank you very much. Thank you.